So I'm going to talk about predicting treatment outcomes using diffusion models. And already, as introduced, I just finished my PhD, and I'm on a 6-month short postdoc, but very fun, with Aviv at Genentech and Jure in Stanford. And I'm launching my own lab just this fall, AI in molecular medicine. And so one of my research areas, or like the big research area, is this holy grail of achieving a personalized medicine. So a setup where really patients receive targeted therapies tailored to their molecular profile. And while personalized medicine is, of course, very broad, let's focus at this particular setup of like cancer treatments, as this is the clinical cohort study I'm going to introduce to you at the very end that has been basically the driving factor of the last couple of years. So I have a patient who has a molecular profile which I can measure very well by now. I have really access to the molecular profile of this tumor, and I want to understand which treatment to give in order to most effectively treat this patient. Meaning, how do these cells change over time in response to the drug? And I want to learn this, understand this, so that if the next patient comes in, right, after this first clinical cohort run, and I can make use of the insights I've gathered to, for the next patient, select the best possible drug in order to most effectively treat this patient. What we have and why this is feasible is a revolution in the omics. So biotechnology methods that really allow us to in-depth measure this molecular profile of a patient. Meaning I can look at the tissue architecture, I can understand the spatial structure, I can look at the microenvironment of a tumor, and in particular I'm able to look at each individual cell and measure many different modalities: epigenome, proteome, transcriptome, genome, and so on and so forth. And by now we are really in a biology of high throughput, of large amount of data, and this allows us to really look into questions such as what is a patient, how is a patient going to respond to a drug, what is the most effective drug to choose, what are the particular biomarkers that allow us to understand the disease. And of course, large data, difficult problem, right? The kind of candidate to solve this is machine learning. But what we see is that traditional machine learning methods, as they are on the market, they don't really excel in this regime. And this has many reasons. Many are actually in the methods themselves. But also, this biology problem gives a very particular setup, very unique, and we are not used to this. Like, this is not how usually machine learning methods are designed. Meaning, I'm only able to indirectly measure things. If I measure a cell, I need to usually destroy it. So if I destroy a cell, then I can only measure it before a treatment, but not after. But here we are interested in really understanding, right, the treatment response of a patient, right? Like, omic technologies are destructive, but what I want to measure is the treatment response. But I cannot measure it. I can only measure before and after. So this dogma of machine learning, of having paired data, like an x and an f of x, the function I learned to predict my y, it's something I'm not given with. Like, I'm not provided with this information. And so this is only one reason, right? But one reason that has been very much the motivation of the methods I developed. So how do we understand, or how do we forecast treatment outcomes? At the core of the problem is really modeling cellular behavior. In silico modeling, how a cell transitions upon treatment. How do cells transition from a healthy state into a disease state? So we need to build machine learning models that really allow us to give them a starting cell in a very high-dimensional space, spanned by all the features we measure with the technologies, is able to understand how these cells are transitioning from healthy to disease, meaning how do the features change upon these transitions. Like, what is the upregulation and downregulation of certain factors? If I give a drug, how is it going to influence? What type of transition do I see into the treated cell state? If I'm able to capture this with my model, I'm able to really understand cellular behavior, and I'm able to really forecast treatment outcomes, understand the roots and causes of disease. At the same time, I need to be able to model work with very heterogeneous cell populations, right? Many different cell states, and I'm able to capture them all in order to understand resistance mechanisms and basically those cells that are responsive to a treatment and those cells that are not. And last but not least, the problem I already mentioned, right? I'm never working with aligned data. So I need to somewhat learn these methods or build these models based on the assumption that I have unaligned data, never a pairing of an unperturbed and a perturbed state. So what is the kind of framework that might help us? It's really this idea of generative modeling. So going away from this classical setup of like predicting a Y of an, like having the supervised setup. Here, like the last revolution of machine learning methods has been really in this like generative modeling field. Given just a random sample of a Gaussian and maybe some condition, we are able to really produce super high quality images. Here, for example, conditioned that the Pope is supposed to wear a puffy jacket. And so this now follows a data distribution. So I'm able to transform a distribution of noise into a distribution of data. The method at its core is based on the assumption that I can just consecutively destroy my image. Starting with a high quality sample, if I'm just adding noise per with like a random non-learnable f, I'm going to end up with random noise. So here just a very brief visualization, right? So I can per destroy the image and generate noise. And now I can invert the process and then go from noise to data. So of course this reversal is not very trivial, right? So because I need access to the score function, but this is something I can parameterize with a neural network. And now I have a very functioning neural network or generative model that produces basically from a noise distribution, data distribution with high quality samples. Of course, we are interested in really understanding and forecasting treated states. And in particular, we are not very interested in just predicting any treated state, but the treated state of a distribution given, right, given my original healthy state or given my disease state, right? So I'm looking at an interpolation or like a stochastic process that describes the interpolation between my mu0 and my mu1, my two cell distributions. And now this becomes a bit more challenging because I cannot just use a random function and add noise. It's not going to get me from one data distribution to the other. I can't really easily sample anymore. It's a distribution that is basically just based on the data I measured. And this is also not a tractable objective anymore. Instead, I need to find somewhat a nonlinear drift that describes the dynamics of my cell distribution from the unperturbed to the perturbed state, from the disease state to the healthy state, and so on and so forth. I can plug this in. I kind of know the solution already, because I want to basically derive the system. I plug this in, and this is now a system. I can slightly make it easier, where I have like two neural networks, a forward drift and a backward drift, that describe the dynamics from one distribution into the other and backwards. And this, in fact, is a very famous system based on a famous physicist, Schrödinger. And these systems are known as diffusion Schrödinger bridges. And what is great about them is that they are now generative models that describe dynamics of cells over time continuously, and we can actually make very nice adaptations to it. So, for example, sometimes actually technologies allow us to record a cell several times, meaning we can look at technologies developed here at EPFL in the De Planck lab, LiveSeq, where we can actually sample a bit of cytoplasm and take from the same cell several snapshots. Sometimes we are also able to look at barcoded cells. So if I find the same barcode in a later point in time, I know it's from the cell with the same clonal origin. But this is only partial alignments. Like these are very labor-intensive methods or very heavily depending on the sampling. So I can use this as like information to encode into the algorithm, but I cannot fully rely on it. But what we did is that we basically built a diffusion Schrödinger bridge based on this. Same system as the forward-backward process I just showed you, just different way of writing it. I'm looking for a stochastic process P that describes the evolution from mu0 to mu1, or here p0, p1. And using these partial alignments, we have now an easy way of developing a robust algorithmic framework using two fairly old ideas, Dup's H-transform and classical Schrödinger bridge theory. So how does it work? We know, right, from the slide before, we can describe this generative model through an SDE where we have some unknown drift, which we need to learn. And because now we have some partial alignments, we know that these cells are conditioned to start at x0 and end at x1. So I can replace here my drift term with this Dupes H transform that kind of describes this coupling. And similarly, for the reference process, the underlying basically base assumption of how this process could look like, if I have paired data, I can actually describe the drift of this underlying reference SDE in closed form. So now this becomes a system that is very easy to learn, very similar to the generative modeling, like the diffusion models that actually have a Gaussian on one end, because I just have a simple regression, like basically understanding how different is my drift from the drift I want that is describing my cellular dynamics. And so now this is again a system that describes continuous time changes of these cell distributions from an unperturbed into a perturbed state, or from a diseased state into a healthy state. And I'm actually making use of whatever the data is providing me, some form of partial alignments. These diffusion Schrödinger bridges are also great because we can incorporate a very important part of modeling biology, which is cell death and birth. So, of course, the prime root, or like the prime purpose of these cancer drugs is to kill cells, right? Or to stop them from growing. So of course, many times I'm actually observing cell death events. How do I incorporate this in this like continuous time process that always operates on certain assumption that I cannot just like lose cells or lose math? We worked on an extension to this, which introduced unbalanced diffusion Schrödinger bridges. And the way we did it is by deriving time reversal SDEs that contain birth and death processes, and thus are able to really model cancer drug effects that are highly induce apoptosis. So basically what we derived is we have this forward-backward SDE, and we derived now this like scheme for systems that have birth and death. So again, right, we can describe our SDE with some like random drift, which we need to learn because we don't know it for the cell dynamics. But now we also have some killing rate that we need to understand. And the killing rate is best actually modeled by introducing something like a coffin state, basically a state where my cells are going to end up like a cemetery the moment they die. I'm also able to model birth this way, because the core idea is that actually time reversal of a diffusion with killing is a diffusion with birth. Meaning now my coffin state, which used to be a cemetery, can be basically a reservoir to sample new cells that in the back all followed a shallow path until they appeared into the system. And so this way we can model birth. So this is very nice because now looking at just cell line data for now, given that we have a system where we sampled after 8, 24, 48 hours, we are really able to model these dynamics that also account for changes in the cell counts. So for example, here we see that there's a certain delay in the effect of the cancer drug. Initially, we actually see high level of birth. Actually, don't find my pointer anymore. High level of birth, right? Like, so actually we see some proliferation until heavy effects of like apoptosis-inducing factors emerge. And so all the death events are basically at the later stage in time. So what does it give me? So I describe basically like these diffusion Schrödinger bridges, why they are great and why they are flexible, right? We can actually incorporate some form of annotation. We can incorporate birth and death. But what do these generative models give us? So let me give you an example of like a previous work, also based on this generative modeling idea, where now the task is that we are provided with a clinical cohort study that has around 50 patients. And for these patients, we actually have the control state, the state of the cells before the treatment, and the state of the cells after treatment. We can use this cohort in order to actually learn a generalized perturbation model, and then use this to understand for an incoming patient never seen before. I look at the cells before the treatment, how the treatment would change the cell profiles, like what type of effects and behaviors do I see? Do the cells respond to the treatment? Do they evade cancer therapy or not? So this is part of this ongoing huge initiative in Switzerland called the Tumor Profiler, where we have like 37 patients measured with a technology called 4i, developed in the Pelkmans lab, and we have 34 different drugs that were screened, so thousands of cells. And so this clinical cohort is very small and also, unfortunately, incredibly diverse. So we have a lot of different subtypes, we have a lot of different driver mutations, right? And they, of course, are all going to influence the response of these patients to the treatment. Beyond the state of the disease before, so the control state, the unperturbed state, all these are, of course, additional factors that are going to influence the response. So here we have a slightly more simple setup where we just try to learn a static map, not a continuous time process, but same philosophy. I learn these maps, these one-step maps with a neural network, and then at test time with a new patient, I basically apply these models I've just learned and parameterized to predict the changes of these cells over time. And what is great, and this is really a factor I want to highlight most heavily, is, of course, we can now predict for a cell and the features, how these features change over time. But most importantly, I can subtract, because now I have an alignment. I have for every cell, I have the state afterwards. And I can subtract these two vectors. And now I really understand these changes in the features. And this is really what contains the information. So on the one hand, of course, I can predict the outcome of an unseen patient. So here we see the distribution of, for example, one p-ERG marker. We know the population of like how it was expressed basically before. So this is for 2 different patients. Before, right, it was actually very heavily expressed or very heavily present in the sample. And then upon treatment, there's a reduction and even like a bimodality that occurs or appears. And the model we developed is able to really capture this bimodality to a certain degree compared to the baselines we look at. But we can do much more than this. We can really look also at the cohort. We have data on how they responded after treatment. We have survival data. How long did they basically still live after the treatment has been applied or after a certain treatment has been applied? Now we can learn, we can look at the models, like forecast their outcome, and based on this, right, this feature response, like how they actually responded, we can predict if they only lived a few days or if the progression was more or less intermediate or very, very slow. So they lived for almost two years, and then afterwards we don't have measurements anymore. And what is interesting to see is that the information is really captured in these changes in the cellular profiles, and not necessarily in just the state of the disease before treatment and after treatment. It also, these methods allow us now to find new clinical associations. So for example, I have all my different meta-information, all these different like driver mutations, biopsies, and so on and so forth. And if I look at the predicted response, I really find much more associations or things that are potentially interesting. And I'm just going to highlight one because I also have only one example for now. This is analysis which is still running. But so here we have, for example, a paclitaxel cell for NRAS mutated patients. And we see that there is somehow clinical association of interest. And this is something we actually know in the literature. We know that this drug enhances microtubule stability, inducing cell death. But patients with this driver mutations, this NRAS driver mutation, actually show an activation of a certain pathway that allows us to evade like resistance mechanisms to this cancer drug. And so this is only possible by not just looking at the state of the cells after treatment, but by being able to reconstruct individual cellular behavior. And this also leads me to the work I'm currently working on. So these are all systems which have been trained and developed on a single data set. But by now, right, around the globe, we have collected so many data sets that we can think or aim for learning really universal representation of these cell states from, by now, actually it's much more, depends if I'm at Stanford or at Genentech, 36 million cells. And I mean, of course, certain companies have much more data than this. And we can not only look at these like small-scale models, as what I showed you in this talk, but instead look at really large foundation models with millions or even billions of parameters. And now I have somewhat a virtual representation of my cell state. And this allows me, or gives me, very new and different means of actually modeling cellular behavior. And the moment I'm able to accurately model cellular behavior, for example, through this virtual cell, which is represented through its DNA, RNA, and proteins metabolome, I'm able to understand or model accurate cellular behavior and thus understand roots and causes of disease and potentially outcomes of treatments for patients. So this is ongoing work, so stay tuned, what comes next. For now, I want to thank the collaborators that have been working with me on this work, particularly my PhD advisors, Andreas Krause and Marco Couturi. So this is a very close collaboration with Lukas Pelkmans and Gabriele Gut. And the diffusion Schrödinger bridges were mostly developed with Yaping, Vignesh, and Matteo. And I thank my current hosts with which I'm doing exciting work. And in particular, if this type of work is very interesting to you, we are hiring. We are part of both the computer science and the life science department. And we are going to work on all these type of methods. Thanks.
Thank you, Charlotte, for a very interesting talk. That was actually exactly very on time. So we can take 1, 2, 3 questions. Yes.
I'm not clear whether you are tracing cells by cell, individual cells, or you are looking at the bulk of cell population.
So we measure single cells. Basically, we sample, like usually we have if we have a tumor sample, we split it in 2. Some of them we measure before treatment, and others we measure after screening with these 34 different drugs. But so these are different cells, but we assume that they are from the same distribution. And this distribution has changed over time. And we are basically able, what we aim for is reconstructing this distributional changes over time. But by doing so, we actually reconstruct individual dynamics. So we have basically predictions for individual cells on the cell level on how they would change over time.
But it is very well known that tumor cells are heterogeneous.
Yes. So
when you split the tumor into different populations, they will have different lineages. So
yes.
It means that when you trace them, they don't necessarily reflect their lineage.
Yes. So basically the assumption is that the moment you split the cells, and some you measure afterwards, some you measure before, that you have somewhat a good representation of heterogeneity in both of your cell samples. Yeah.
Thank you so much. That was a really exciting talk. I may be a little bit related, but I'm wondering if you can help us develop an intuition for what size of data set you might want to go for if you have these partially paired samples, especially in terms of number of patients. How do you think about that?
So the partially paired, so this was a data set where we have like around 5,000 cells. So this was so the experiments were all run on barcoded data. The thing is, you can easily combine these 2 methods. I can basically learn a diffusion Schrödinger bridge on all unpaired data and I can basically learn a prior on the few hundreds I have that are paired to give me a good form of reference process, right? I was always showing you this reference drift. I only touched upon it, but you can basically develop a prior on how the dynamics look like because by default, prior is just Brown in motion because you have no intuition. You can update this with a few hundred cells to basically capture the type of dynamics. In particular, if you are able to sample rare cell types, right? You can basically prior that this is something that shouldn't disappear in the bulk. And then you can use these high throughput methods that are not barcoded or that are not live-seq and basically fine-tune the process or train to the end, basically.
And so for number of patients, for the paired data, you don't really think of that as necessarily as super important?
So, I mean, what we see here, 37 is not enough, right? So I didn't go through the analysis, but I can show you exactly, and this is also a learning process, right? I can show you exactly for which patient I can make predictions and for which patients I cannot make predictions. And so currently we also work on extensions that basically flag this, right? So that we have an idea, is this a prediction I'm certain about or not, right? So 37 is not enough, but if you think a bit different from a machine learning point of view, and you don't really think in terms of like number of patients, but like number of cells, then these methods actually provide us quite a bit. And in particular, at the very end, right, when I was talking about these methods, if you go in this philosophy of these like virtual cell representations, like when I'm able to, like I have basically learned models on all 33 or 200 million cells, right? And now I come with my small data set. But if I'm able to basically map it into the space and gather experience from all the things I've seen so far, then I think this becomes less and less and less of a concern and a worry.
Thank you. One last question. Make it short, please. Sorry.
Okay, thank you for the presentation. So, if I understood correctly, the last part was the optimal transport part, and the first part was the Schrödinger bridges.
It's also optimal transport, actually. But,
exactly, that's my question. So, they look quite similar because in both cases you try to find couplings between an initial state and then the target state. But I didn't understand why you use you develop both of these methods, because in the first, I guess in the first part with the Schrödinger bridges you have continuous time somehow, which means you have a lot of measurements, or you create by adding noise. I'm not sure.
Yeah, because machine learning research is faster than research in biology. So it's these methods are from this year and last year, right? I can train and test them on the cell type of cell line system, but I mean, the very first method we developed, there was a certain, like, somehow selection process. What's the best methods to choose to really, like, move to this clinical cohort study? So this we started 2021, so basically at the beginning of my PhD. And so we are still on it because now there's a lot of, like, biological findings, like very different type of research. Machine learning, like, we develop methods that's much more fast-paced. So the diffusion methods, they are very, very powerful. We also know this from the image generation. They operate better on very high-dimensional data. They have higher sample quality, and continuous time is interesting, in particular when I'm looking at diffusion methods where I can really understand and steer the path, right? Optimal transport is great, but not everything in biology is the shortest path, but I have much more leverage here. So this is very active machine learning research, right? These methods don't exist.
So you started with optimal transport and then you transited to the Schrödinger?
So I mean, Schrödinger is still optimal transport. It's entropy regularized optimal transport, but you can actually move to any type of, like, optimality is a flexible word, right? So depends on optimal against what.
Okay, yeah, very interesting. I will